The T cells of your immune system are your built-in security guards. They surveil your entire body, seeking out and destroying cancerous cells. Most of the time, they do a pretty good job. But as Matt just explained, sometimes cancer cells can slip by undetected. So we have a tool that helps us engineer T cells to be better at detecting cancer. We can take a patient's T cells and fit them out with a receptor that acts almost like an antenna, locking in on cancer-specific markers and directing the T cell's tumor-killing machinery against them. These receptors are called CARs, and CAR T cells have been incredibly effective against blood cancers like leukemia. But I work with a deadly subtype of pediatric brain tumor, and that's an entirely different story. The bloodstream is the natural habitat of the T cell. The brain, on the other hand, is more like a barren wasteland, difficult to get to and almost impossible to survive. Oxygen levels are lower, other immune cells that would typically boost the T cell's performance are absent, and the tumor itself sends all sorts of signals that weaken T cells. CAR T cells are incredibly activated, but activated T cells are also more likely to enter a state of exhaustion, where they can't perform their function anymore. CAR T cells are sprinters, but in the hostile environment of the brain, you need to be able to keep your pace. So my lab is working to improve CAR T cells for brain tumors by putting the control of the CAR under a genetic switch that detects a brain-specific protein and then turns on the CAR. I like to think of these cells as biological sleeper agents. When I inject them into the bloodstream of a mouse, they're completely normal T cells until they travel to the brain, hear their code phrase, and activate into tumor-killing machines. At first, the idea was just to make sure that these cells were only active in the brain, and not targeting any other cells and other organs that just happen to look like tumor cells. But we quickly realized that by delaying the activation of the CAR, we were essentially allowing the cell to save its energy until it got to the brain. These inducible CAR T cells are stronger and less exhausted. They clear tumors faster and keep them gone longer. When I look at the brains of these mice weeks after injecting T cells, I can still see those inducible CAR T cells sticking around. These security guards are not sleeping behind the desk. They are alert and energized. My lab is currently working on a clinical trial to test these cells in adults with brain cancer, and I'm very hopeful that my research will help pave the way towards testing in pediatric patients as well. There is a huge need to find safer and more effective treatments for brain cancer. If we can harness and control the natural tumor-killing power of the T cell, we can bring new treatment options to the patients who need them the most. Thank you.